All right, hey everybody. Welcome to another episode for your AP Biology notes. So we're going to be going over two concepts today. We got 6.1, which is going to be all about DNA and RNA structure, just so that way we can go ahead and go through those guys real quickly. It's very similar to when we went over DNA and RNA during 1.6. So if you want to go ahead and just flip through your notebook real quickly just to take a look at that, it's really going to be almost like a review of that same information, just going a little bit deeper. I still want you to go ahead and build this into your 6.1 notes, but just so that way you'll be able to refresh yourself and see what we're going to go take a look at. And then afterwards, we'll go right into DNA replication. So let's get into it. Here are your essential knowledges. It's just basically, talk about your DNA. How is DNA slightly a little bit different in prokaryotes? And then just talking about our base pairing rule. So let's go and revisit nucleic acids, right? Back in 1.6, when we were talking about all of our different biological molecules, we mentioned nucleic acids are used for the storage and transmission of genetic information. And this is basically used for everything. So during that time, we ended up talking about DNA and RNA. And that's actually what this whole unit is about. It's specifically like, what does DNA look like and how do we make more of it? What is RNA? How do we use that and make more of that? And then basically, how do we like regulate these processes, right? Or how can we end up changing them? That's kind of like the whole idea about this. So it's really going into very, very, very deep detail about stuff. So DNA is typically like your normal thing that we're going to go use for genetic information, but there are some cases that we can end up using as RNA as a form of heritable information. Specifically, we can end up seeing this in viruses. Notice that your virus right here, right? Viruses come in like different flavors. You can end up having a virus, just with their little spike proteins all over the place, right? You can have DNA that's located within there, and how viruses work is that basically your virus is going to go ahead and enter a host cell, right? Because the host cell has a receptor protein that binds to the spike protein. And it's like, oh, hey, you must be friendly. So we're going to go do a handshake, and then allows the virus to get in. And then once the virus is in there, it's going to go ahead and more or less hijack the host's DNA replication stuff in order to go and make more viruses. Basically, makes more DNA using the process we're going to take a look at with DNA replication, and then also makes like the protein coat and these antigens by doing the translation process by using the ribosomes. Now, the reason why I bring this up is that you have some really, really interesting viruses that are out there that we can call retroviruses. Retro, right? So in a retrovirus, it actually uses RNA. In the case here, and an enzyme called reverse transcriptase. Reverse transcriptase basically goes from RNA and we reverse transcribe, or basically we turn that RNA back into DNA. And then from there, we're going to go ahead and, through transcription, like normal, we'll make more RNA. So there are some cases that you can actually use RNA as your primary source of heritable information. Most of the stuff that you're going to go take a look at is mostly always going to be DNA, but don't ever count that out. RNA can still be used. There's currently some theories and debates in the scientific community that, if we go and take a look at the, LUCA last uniform, common ancestor, basically the first living thing that's ever existed, right? It's basically a cell, a really, really basic cell, right? That just has like a cell membrane and it has to have some kind of genetic material. And basically, the cell has to do two things. One, it has to survive. And then secondly, it has to be able to replicate itself. And what scientists are kind of like talking about right now is that they believe that the first cell that ever existed was actually like an RNA cell. One, right, because you're basically using like half of the amount of genetic material, right? It's only one side of a DNA ladder, right? So it is a single helix, so it's probably a lot easier for you to go ahead and throw a plasma membrane around that, and then through evolution, natural selection, right, just random chemical processes, you end up getting the correct enzymes, in order to go and start to replicate yourself, and then you make more and more and more these cells. And then probably over time, we ended up, something happened where you got to go to get DNA, which was more regulated, more special, and all this other cool nonsense is happening. Now, the reason why we're focusing on this is that the genetic information that we have is being passed, that's stored and passed to later generations, okay, through this DNA. So in the case here, you have your cells, right? Here's your DNA during the, S phase of the cell cycle. We're going to go ahead and replicate it. And then from there, we can end up passing along with this genetic material to the next generation, right? So this is showing us meiosis. We're having crossing over take place, getting a lot of genetic diversity, right? But this is important, right? So the DNA that you have right here, this DNA like started from somewhere. So if we go back like many, 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 millions of years ago, right? Like it's been using the exact same genetic information, just mutations have been occurring, chromosomal mutations have been occurring, and lots of other very interesting events have taken place in order to make all the diversity of life as we have right now. But I am seeing the genetic information, right? It's passed on to all these generations. So, we kind of like play an idea about evolution for a second. You know, here is, here is you. And let's say here is a blueberry bush, right? Now, this blueberry bush is great. It's awesome. It's wonderful. And if I go and take a look at the cells at the blueberry bush, guess what guess what organelle I'm going to go ahead and find within my blueberry bush? I'm going to find mitochondria. And if I go into my human and I go and take a look at its cells, I'm also going to go find mitochondria. And not only do I find mitochondria, I even find like, the enzyme that creates all the ATP, right? I see ATP synthase. So this human has ATP synthase. This blueberry bush also has ATP synthase. Well, why is that? Well, at one point they had some cellular common ancestor, and just through the tree of life, they evolved into what they are now, and that's why we have our blueberry bushes and our humans. We have the same DNA that codes for these same proteins, right? We're passing on this genetic information, right? It's like millions and millions and millions and millions of years of just this stuff being passed on. It's incredible. You go take a look at family pictures, and you can see that how you look like your parents or your grandparents or even your great grandparents. Well, they're passing along that genetic information. Those traits are going to you. This is why they say like you got your mother's eyes or you got your grandfather's nose, because you're actually passing along those traits. They can even be bad traits, right? They can even be things like potentially you have cancer causing cancer in your family, right? Same idea. You're passing along that genetic information to the next generation. You know? All right. So with that, let's go ahead and go to the next one. Now, what makes bacteria a little bit interesting about DNA, and specifically, it's how the chromosomes are set up. Okay, so prokaryotes, right? If you think about eukaryotes, right? Eukaryotes versus pro. So here's your prokaryotes. Here is your eukaryotes, and we're taking a look at DNA, right? We both have chromosomes, right? But here's something that's interesting. Bacteria have one chromosome, right? They have one circular chromosome, right? So their chromosome looks like this. While in the case of a eukaryote, right, we have the chromosomes, right? It's all this like chromatin-like material, right? And it's located within the nucleus, right? The nucleus. So, and it depends on the organism, right? Humans, we have 46 chromosomes, right? These guys only have one. Right? That's a huge difference. And then also there's a difference in that because they don't have the nucleus, they have a nucleoid region, which just basically means this is where the genetic information is. But something unique that bacteria have that we do not have are these really cool things called plasmids. Now, what a plasmid is that a plasmid has the ability to basically incorporate itself or be removed from the bacterial DNA. And this is actually kind of how like a bacteria can regulate. It can actually regulate what genes it wants to go ahead and express. There's some internal communication that takes place. We're going to go make a plasmid, take a plasmid out, and it is what it is. Now, what's neat about these plasmids is that if you had another bacteria over to here, bacteria have a really interesting ability where they can end up using what's called a sex pili, right? And an idea about sex, right? What do you do in sex? You end up transferring genetic material. It can actually transfer this plasmid over here to those other bacteria. It doesn't even have to be the same species of bacteria as long as like it's able to stick onto it, right? And then the bacteria ends up becoming transformed. We're going to be talking about transformation later on in this unit, but this is actually something unique to the plasmids. And when we do our bacterial transformation experiment, this is actually what we're going to be playing with. So don't sleep on this, all right? You definitely want to make sure you are aware of your plasmids. This is just the information that we mentioned about your eukaryotes, right? They have more than one chromosome. Their chromosomes are linear. They're in like lines, and we find them in the nucleus. We're not going to go into more detail about that. It's not necessary, right? I already mentioned about your plasmids. You can go ahead and take a pause to it, but what you can end up seeing is that this is where how the plasmid can end up being located. So what ends up happening, and the way to think about it, right? So here is your wow, I should. 1 second. Everything has to be circles. 1 second. All right, so here is your bacterial DNA. And then let me do another color for the plasmid. So I'm going to do the plasmid as like this purple color, okay? So here is your plasmid. And what happens is that it just incorporates into the DNA. So then what I'll do is I'll just kind of draw what this ends up looking like after we end up incorporating the plasmid in here. And now it's part of the bacterial DNA, right? You can see that the plasmid is now injected itself into the original DNA. Now what that means is that I can now start to transcribe and translate that specific gene now. Right. And that could have been a plasmid that I got from another bacteria, right? It could end up making it that your bacteria mutates and it becomes more virulent or something like that. This is how it can end up becoming more virulent. All right, so just to round this off, your purines and your pyrimidines, you just want to go ahead and keep track of which one is which, as well as your base pairing rule, where you have your thymine will bind with either uracil or adenine. Uracil is specifically in RNA, while adenine is in DNA, and then cytosine is always with guanine. And your difference there is how many hydrogen bonds forming with them. This is important because of the intermolecular forces, right? Keep in mind, don't ever sleep on those IMFs, right? Because of those intermolecular forces, that allows there to have the 3 hydrogen bond here's or the 2 hydrogen bonds over to here to allow them to actually pair up, right? Because if I go and take a look at my DNA ladder, every A pairs with a T, every T with an A, every G with a C, and a C with a G, right? In the case of us talking about this over to here, let's say that I start to transcribe a DNA. Let's see if we remember this. I'll do ATCG as my DNA sequence, right? And then I'm going to go ahead and change it up to maybe this color right here in order to do RNA, right? When the RNA starts to be transcribed, the A will pair with a U, the T pairs with an A, C with the G, and G with the C. And then this would end up going off to the ribosome and being red, right? So just making sure we knew this real fast. Right. And then here is your information with your purines and your pyrimidines right here, just making sure you can identify which one's which. Your purines are going to be the ones with the 2 shapes, like the hexagon, pentagon shapes within them, while your pyrimidines are going to be just like that hexagon shape with it. Again, they are designed this way in order to match up, so that way the hydrogen bonds work properly. And so this should have been a review. We are going to roll right into 6.2. All right, so 6.2 is all about how DNA gets replicated. This is everything that comes from the book. We're literally just going to go through this whole entire essential knowledge bit about this. Please take a look at the exclusion statement right there. I'm going to make sure that you are aware of this. You just need to know the enzymes that are in there. We are going to make sure that you see these terms. I'm also going to go mention Okazaki fragments. It's not mentioned in here. I would rather you just be familiar with what they are. So that way, when I mention it or if you see it, you're just like, what is this Okazaki fragment? And you'll be all right. Okay, so first of all, let's go ahead and do a quick thought experiment, right? DNA is necessary for all cells to have the correct information after undergoing mitosis, right? So if we think about this for a second, right? If I ended up having my cell, and this is my cell right here, right? So I got 4 I have 4 chromosomes. If I go through mitosis, right, I'm only going to get like half the DNA, or I'm only going to get like 2 chromosomes in one, 2 chromosomes in the other. It's not going to go work. So what's necessary is that we are going to need to always copy the DNA first, so that way I can go and have, right, I can have a chromosome in each specific one, and now I'm all squared away, right? If it's not copied correctly, you might be missing that key information that is necessary for the cell to be able to survive. All right. Now, our whole goal is basically explaining this picture right here. If you can tell me what the topoisomerase does, what the primase and the helicase do, if you're able to tell me about the RNA primer, DNA polymerase, being able to identify the difference between a leading and a lagging strand, you are going to be okay. The Okazaki fragment, right? This one isn't mentioned for us to go and know about. I'm going to give you some heads up about it so that way when you see it, or if they mention something about it, you have the background in order to be successful with it. Now, the first thing that we need to discuss before anything else with this is the direction of DNA replication. In this class, in biology, stuff's done 5 to 3. Everything is done 5 to 3. Everything has evolved to do everything 5 to 3, 5 prime to 3 prime. And if we do not remember, 5 prime to 3 prime just mentions which carbon we're talking about. Are we talking about this carbon here, this one, this one, this one, or this one? 5 prime to 3 prime means that we are going down in this direction from the 5 prime to the 3 prime. So everything goes in that direction. All right, everything is done 5 to 3. Everything is doing 5 to 3. Now, let's go ahead and take a look at our picture, because this is where I'm going to start to break your brains a little bit, okay? We have DNA. Keep in mind, right, your DNA is anti-parallel. So it's 5 prime over here and 3 prime over there, but it's 3 prime over here and 5 prime over there. Take a look at our image, and I want you to take a look. What direction is the DNA replication happening? It's going this way, right? It's going this way. So we are going 5 prime to 3 prime. So I am copying the DNA going this way, but I'm traveling this direction. So I'm traveling this direction, and I'm copying it 5 to 3 this whole time, right? But the problem here is that because this 5 to 3, so I'm doing that over to here, but since this area, right, because of it being opposite, right, I still need to do 5 to 3. So how am I able to go and get it done 5 to 3? Let me go and take a pause for a second, okay? Let me take a pause for a second, because I feel like I'm going to go in and throw you all off, right? Okay. The 5 prime to 3 prime is over to here. All right, so it's going in this direction. Everything is being done 5 to 3, right? So here's the 5 over to here. Here's the 3 over to there. It's moving in this direction over here. We are going this way, all right? We're going this way, right? So we can see that here's the 5 prime, 3 prime. We're going that way. We're moving this way. But if I'm going this way, right, notice that I would, here's the direction right here. But if I'm going this way, I still need to copy everything 5 to 3. I still need to copy everything 5 to 3. But the problem here is that if I'm going this way, I can't, this is going to be like continuous. This is not going to be continuous. So this is what causes the leading and the lagging strand to show up. So this is causing the leading and the lagging strand to show up. The reason why I call this leading strand is that this is going to be continuous, but this is not going to be continuous. And this is where these Okazaki fragments show up. It's that basically it's saying that every little tiny section of DNA has to kind of be glued together. So I'm going to go stick these Okazaki fragments together to eventually create my full DNA sequence. If this is weird, I'd say watch a video. I'm going to have a video available for you to go take a look at so you can kind of see it. And if it's still confusing, ask your questions. We'll make sure you're all squared away. All right. This is probably the hardest thing for people to kind of wrap their head around, that everything is done 5 to 3. And that's why this can be done continuous while this one is the other one's going to be done backwards. DNA replication is semi-conservative. What that means is that you, at any point in time, are going to be keeping half of the DNA that you started off with. So if I take a look right here, right, I have this red one right here and this red one right here. If I go and take a look at my following generation of DNA, right, so this is after one split. Here's my red that I had from the first time. Here's the red from the other one, all right? So I only made half the DNA for my total DNA, right? It's semi-conservative. I am conserving part of my DNA. Then if I were to split this again, I can see here is the red right here. Here's the red right there. Then I have the blue, right? So the blue is from here and here can be located right there and there. So what I need to do is create a new DNA sequence so that way everybody ends up having their own DNA. There is an experiment that they used to go and talk about. We don't really talk about it anymore. Basically, what they did was that they took I think radioactive markers and to try to see where the DNA was. And then originally they thought that it was not conservative. Like basically you'd have like two purples and like two blues, right? So like it'd be like blue, blue, and then you'd have like purple, purple. So like the purple would just have stayed like the same. The heck, is this not working? That's fine. I don't know why my colors are not working. But anyway, they found out that it was ending up being a semi-conservative process. Whoop. All right. And then for the actual copying process, this is everything that ends up happening. You can go ahead. I would recommend that you write this stuff down. And all I'm going to go do is just pull it up on the bigger picture and then just explaining all these little bits right in there. Okay. But the helicase unwinds the DNA. You have topoisomerase relaxes the supercoil. Really, topoisomerase should probably be the first thing that ends up taking place here, right? So this is like step number 1 right here, while this is more so step number 2, because you can see right here, here's topoisomerase right here, and then here's the helicase. Then the DNA polymerase is going to need an RNA primer. Another reason why we think that RNA was probably the original molecule used for all genetic information, because we use RNA primers in order to make this happen. So you're going to have some kind of RNA primer right here for the DNA polymerase to latch onto, and then it's going to go ahead and synthesize one DNA will only synthesize one half of the DNA. So one of them will be continuous, which is your leading strand, and the other one's discontinuous, which is going to be your lagging strand. So here is your lagging strand right here, made up of Okazaki fragments. Here's your leading strand where it just consistently is making a DNA, not having to remove itself. And then finally, we're going to be using ligase in order to go and stitch everything back together. DNA ligase is order to stitch the Okazaki fragments all over. That's basically it, everybody. That is the process right here. So when we are going through the S phase, right, keep in mind, this is the S phase of the cell cycle. When we decide to replicate the DNA, right, so you have your 46 chromosomes. When your DNA, your chromosomes, go from this to this shape, right, this is actually what is happening. So we start off by relaxing the supercoil by using topoisomerase. We then use the helicase here in order to actually break the hydrogen bonds that are holding the DNA double helix together. And then from top side, right? So here's the 5. So you got 5 over here, 3 over here. This is just like all this stuff is going on over here. Okay. So from here we have to do 5 to 3 over to here. So from here we're going to be having to do everything 5 to 3, but it's going to be backwards. So we're going to have to go and start it at multiple different spots right here in order to go ahead and create our DNA sequence. That's where the Okazaki fragments come into. This is where we use the DNA ligase in order to go stitch everything back together. Your leading strand, on the other hand, right, that is continuous. This DNA polymerase just hangs out over to here. It's going to go ahead and consistently be building. It's going to be consistently copying down the DNA, and everything is going to be hunky-dory. There's a little bit more stuff that goes along in here. The animation is going to kind of show you like why it is that the Okazaki fragments end up being formed and how they all end up getting stitched back together. So come with your questions, and I'll catch you on the next one.